Hello, good morning again. We are going to start. Back there, please sit down. So we've made a slight change in the program. We are going to continue with Andrew Salter to try to avoid computing delays, making all the changes of the computer and all the slide presentation. So he's going to talk about fetal programming and fetal programming of lipid and lipoprotein metabolism. What, why, and how. So I'll let you with him. Thank you.
Okay, thank you very much. Sorry you've got me again, but at least you've got a very different topic this time. So more about the person now rather than what they're actually eating. So I'm going to say a little bit about fetal programming, something that I'm relatively new to, but there's a lot of expertise in Nottingham and a lot of people in Nottingham who are involved in it, who I will credit at the end. So I've called it fetal programming. My colleagues who work in the area don't like the word programming for some reason, and they prefer to call it the developmental origins of health and disease, or DOHAD. And it's one of the few areas in recent times that the UK can actually claim for its own. The ideas really did come from the UK, but not from Nottingham. They originated in Southampton, and they really originated with the work of David Barker. And he came up with the fetal origins hypothesis, which is often referred to as the Barker hypothesis. Finding a definition of it is more difficult than you'd realize, but the one I've taken is directly from David himself, where he says fetal undernutrition in middle to late gestation, which leads to disproportionate fetal growth, programs later coronary heart disease. That's how it started. The whole field has got massively bigger, as you'll see, and relates to a whole range of different diseases. But that's where the work really started. It's certainly more than just coronary heart disease now. Stroke, obesity, metabolic syndrome, type 2 diabetes. There's good evidence, human epidemiological evidence of all of them potentially being programmed while you're in the uterus. And there is emerging evidence for other diseases such as cancer and even schizophrenia. So it really is a large range. A little bit worrying that potentially you may already have your susceptibility to these diseases being programmed into you, not by anything that you've done, but potentially by things which your mother or even father might have done during the time that you were conceived. So you can blame them rather than yourselves in future if you want to. Many of you will be aware that the classical key findings which started this whole thing emerged around the findings of what happened during the so-called Dutch hunger winter, when the Germans put siege to the population, and indeed for several months they were on very, very low calorie intakes, virtually starving, stories of eating tulip bulbs and candle wax and all sorts of things. And inevitably during that period, there were women who were pregnant, and there were women who were in different stages of pregnancy. And somebody had the foresight to actually follow these people up, or the offspring of these people up, and actually see what had actually happened to them. And it was found that those people who were in utero in early gestation, in later life, and we're talking when they were 40, 50 years old, had increased obesity. They had a more atherogenic lipoprotein profile. That's what I'm going to talk about. They had a higher LDL to HDL ratio, and they had increased mortality and morbidity from coronary heart disease. Those who were already well through their pregnancies, their offspring, those who were exposed in late gestation, seem specifically to be more vulnerable to impaired glucose tolerance and risk of developing type 2 diabetes. So, through this accident of war, through this starvation, a relatively short period, so the women survived, importantly, the pregnancies went all the way through and the children were produced, we got some of the first evidence of potential programming of chronic disease in utero. David Barker followed this up by looking back in historical records which were kept by some of our hospitals, and the Hertfordshire cohort study is one of the most famous. These were very well kept records from, 60, 70 years ago, and they seem to show more or less the same thing. So the graph here shows the standardized mortality ratio from cardiovascular disease below the age of 65 according to birth weight. And as you can see, apart from the very heaviest babies here, there's quite a strong tendency for mortality to go down as birth weight increased. So those children who were the lightest tended to have the highest level of heart disease. Those who were the heaviest had the lowest level of heart disease. When you get above about 9.5 pounds, then it seems to start to reverse. Often, for example, the mothers of those children may have been diabetic themselves. That may have had an influence on it, as that's often associated with having babies of high birth weight. That was true in women, and it was also true in men. So separate evidence from the Dutch famine that potentially what happened to you in utero is having an impact on your health later in life. And finally, from the epidemiological evidence, this is just looking at the lipoproteins, the HDL to LDL ratio in tertiles. So this is the highest, this is the lowest. And again, you can see that the distribution of LDL to HDL is very different depending on your birth weight, the lowest weight, medium weight, and the highest weight. So a range of epidemiological evidence to suggest that certainly birth weight is associated with coronary heart disease, and perhaps through that, your 9 months in the womb or whatever is actually having an impact. There's lots of other epidemiological data, including some in Asian people in India, which I might mention towards the end, which fits in with our first talk today. This was followed up by a mass of animal experimentation. And really my colleague in Nottingham, Simon Langley-Evans, was one of the first to actually demonstrate specifically in an animal model, in a rat model of low protein feeding, that you can actually program hypertension, for example, by feeding the mother a low protein diet. Since then there's been a whole range of different studies. We've got studies in rat, sheep, in mice. Most of these have related historically to either severe nutrient deficiency or specifically protein deficiency. In the last few years, more and more studies have started to look at overnutrition. I'll say a little bit about that at the end. And the disease endpoints which have been looked at include blood pressure, plasma lipids, obesity, and insulin resistance. And in general, most of these studies have confirmed that there are critical periods during fetal life where a nutritional insult will program metabolic disease factors later in life. This is one of my own studies, and it must be an old slide because that paper was published about 2 years ago now, but this was a study again using our apoE-Leiden mouse. Our model of increased risk of atherosclerosis. But this time we took mothers and we fed them either a normal diet or a normal diet which was adequate in all nutrients but low in protein. So the normal diet I think had 18% protein, the low protein diet had 9% protein, but all the vitamins, minerals and everything else were in there in normal amounts. We then took the offspring, and those offspring were then raised either on a normal chow, a normal mouse diet, or what we call the atherogenic diet, a diet specifically enriched in saturated fat and cholesterol. 12 weeks later, we killed the mice, and we looked at our lesions in the same way as I was describing in the first one. And again, here we have a section through the aorta, just where it joins the heart. These little bits here are the valves, and all of this red staining around the edge is actually the development of atherosclerotic lesion. On a normal diet, this mouse model doesn't develop any atherosclerosis, and it didn't matter whether it was a low protein or the control mothers they came from. But when we moved to the atherogenic diet, you can see that our controls, so these came from mothers fed a normal 18% protein diet. We had an increase in atherosclerosis, but in those that came from the mothers on the low protein diet, you can see that the amount of atherosclerosis almost doubled. That was when I first started to believe. I was incredibly skeptical about this, I have to admit, until about 3 or 4 years ago. I didn't really believe in it. I didn't think it could be true. But when I saw that data, I really had to start changing my mind and believe there was something in it. I'm not totally convinced that all of the answer lies in lipoproteins, but there was a programming of both triglyceride and cholesterol levels in these animals. Again, the offspring were kept on this low-fat, normal chow diet. They had very low levels of total cholesterol. We've actually got our control wild-type animals here, as well as our apoE-Leiden, because we did it in both the atherosclerosis susceptible and the non-susceptible. If you look at the black bars, they're the most important. So very low levels on our chow diet. Cholesterol goes up dramatically if we feed the offspring on an atherogenic diet, and it went up more significantly in the offspring of low protein. Now, whether that difference is really enough in itself to explain the increased amount of atherosclerosis, I'm not convinced, but certainly there is some programming there. Similarly, triglyceride was significantly higher in the offspring from low protein mothers fed the atherogenic diet compared to those from mothers who were fed a controlled diet. So we had apparently programmed these animals to get more atherosclerosis. We have at least partly explained that by an increase in their plasma cholesterol and possibly triglyceride as well. $1 million question is why? Well, basically, what we believe, or what most people will accept, is that the mother will essentially do all it can metabolically to try and ensure that that pregnancy gets through to full term. And the developing embryo will do everything it can to try and ensure that it gets through to full term. So when you have a nutritional insult, there will be changes occurring in this developing fetus, which will allow it to survive maximally. You then produce a baby, and often the only different phenotype, but sometimes not even evident, is a lower birth weight. That baby will go through life, and my own kids, I always put that on just to embarrass them, because they know there's a picture of them up. They go through life, they grow up with no apparent sign, a relatively silent phenotype, until they reach their 40s, 50s, 60s, and then you start to see an increased risk of development of chronic disease. Now, some people have taken this one step further and called it the thrifty phenotype. They've suggested that because this baby is in a environment where it knows, metabolically knows, that there is a limited amount of nutrients around, its whole metabolism changes so that when it's born, it is well adapted to actually live in an environment where there is a limited amount of nutrients around. But when it's actually born, it suddenly finds itself born into a world with McDonald's and Burger King and Pizza Hut and everything, and it can't cope with that, and that's why that happens. I think it's fair to say that the fetal programming world is divided over this. I don't believe that. I believe it's all about the changes which happen to go on here to try and make this embryo survive until birth, After that, it's just an unfortunate accident that the changes which happened in birth give it disease later in life. And the reason I believe that is we don't see these diseases until well after reproductive age. There's no genetic evolutionary pressure to build that into the program. Obviously, historically, we were built to reproduce as soon as we reached sexual maturity, long before we'd have developed heart disease or hypertension or any of that. So I just think this is just an unfortunate accident, a consequence of what's going on here. But there will be people who tell you it's this thrifty phenotype preparing the offspring for a world where nutrients are limiting, suddenly being born into a world where there's excess nutrients. I'll let you decide which one you believe. So that's just my arguments there. Evolution requires us to live long enough to reproduce. The fetus adapts to uterine environment to survive to be born. That offspring will live long enough to reproduce. After that, it's on its own. We don't have any evolutionary pressure. That's why we have heart disease. That's why we have cancer. That's why we have all these chronic diseases, because we've already reproduced. They're never going to be actually evolve out of the human race. So that's what it is. How does it happen? Well, this is another study that I was involved in with, again with Simon, and this is a different model. This is our rat model of low protein, and the rats were, pregnant mothers were restricted in protein either early, mid or late pregnancy, or the final group throughout their pregnancy. And then we looked at the offspring at different times. We looked at the offspring about 1 month after birth. We looked at the offspring at 9 months after birth. And unusually, we looked at the offspring at 18 months after birth. Very few people take these studies that far, practically because it's very expensive to keep rats that long, unfortunately. But we did do it this time, and we're certainly glad we did because, as you'll see in a moment, it had a marked effect on the results. So different levels, different times of exposure to low protein when the mother was pregnant. And then these animals were just all on a normal low-fat diet, killed groups at these 3 different times. And this is why I'm so pleased we kept them for 18 months, because when we looked at these rats, we found that after a month we measured their plasma cholesterol, there was no difference. This is young to middle age for a rat. Looked at their plasma cholesterol, no difference. This is getting on. This is midlife to old age, 18 months. A rat maybe lived 2 years. And as you can see, by 18 months we got this marked difference in plasma cholesterol. So something that happened in the uterus 18 months ago had no effect on the animal when it was 1 month old, had no effect on the animal when it was 9 months old, but wow, what a big effect when it was reaching old age. There was a whole range of parameters which changed, even more dramatically. Again, plasma triglyceride, nothing at 1 month, nothing at 9 months, 3 to 4-fold higher in the offspring at 18 months. And it wasn't just the triglyceride in the plasma. If we looked in the liver, I'm sure you've heard that the latest epidemic sweeping around the world is non-alcoholic fatty liver disease, NAFLD, the accumulation of fat in the liver. A silent disease for a long time, but a proportion of people who've got it will develop hepatic steatosis, NASH, which is non-alcoholic steatohepatitis, I can never say that, liver failure, cirrhosis, all of those things in a proportion of the people, associated with obesity and associated with insulin resistance. And as you can see, by 18 months, our offspring of the mothers on low protein had severely increased lipids, the little white dots you can see on there, the actual lipid droplets which are accumulating in the liver. So we programmed a whole range of factors which would relate to their risk of disease. We programmed an increase in cholesterol, we programmed an increase in triglyceride, and we programmed an increase in lipid accumulation in the liver. We then went on to look at the changes in genes, gene expression in the livers of these animals. And we were interested in 2 in particular. One, steroid regulatory element binding protein 1C, I think those of you who listened to my talk 3 months ago know that that's one of my obsessions. And the other one, PPAR alpha, as I'll say a little more about in a moment. This one is particularly involved in lipogenesis, the synthesis of fatty acids. This one is involved in fatty acid oxidation, potentially in the liver. They're both transcription factors. They regulate gene expression, but they regulate different genes. So here we have the SREB1C. The black bars are the 9-month-old animals. The white bars are the 18-month-old animals. We have the controls. We have the different stages of restriction at different stages in pregnancy, and we have those which are restricted throughout the whole of pregnancy. First thing which surprised us, which we hadn't realized, was that there are natural changes which occur in a normal rat as it ages. Compared to 9 months, by 18 months, there's a marked decrease in the expression of SREB1C. But the whole programming, that might be a natural program if you like, seems to be disrupted in our offspring of mothers fed the low protein diet. Doesn't matter when, but basically at 9 months they had much lower levels of this SREB1C, but by 18 months this had gone up quite dramatically. If we look at PPAR alpha, you'll see in the controls, as the animals got older, PPAR alpha actually went up, while in the offspring of the low protein animals, it actually went down. So there seems to be a natural programming with aging, such that SREB1 goes down and potentially PPAR alpha goes up, which could regulate the whole of your lipid metabolism, which is completely disrupted and almost reversed in our offspring from the low protein mothers. So what SREB1 does, it's a member of this family of steroid regulator element binding proteins. SREB2 is involved in regulating many of the enzymes involved in cholesterol synthesis and the LDL receptor. SREB1C, on the other hand, is involved in regulating many of the genes involved in fatty acid synthesis. So it regulates acetyl-CoA carboxylase. It regulates fatty acid synthase. It regulates our friend SCD there. And when there's high levels of SREB1C, it binds to the promoter of these genes. It increases the expression of those enzymes and potentially increases lipogenesis, the synthesis of fatty acid. SREB1C goes down. It does exactly the opposite and reduces lipogenesis, reduces the synthesis of fatty acids. And what I meant to point out on here is we also looked at the... They're not on there. Must be on the next slide, sorry. On the other hand, PPAR alpha is another transcription factor, a so-called non-steroid hormone activated receptor. It's activated by fatty acids and their derivatives, expressed in the liver, and primarily regulates genes involved in fatty acid oxidation, so the breakdown of fatty acids rather than their synthesis. It's particularly pronounced, its activity in rodents, because it induces the proliferation of peroxisomes, and these peroxisomes are specifically involved in oxidizing fatty acids, and you get a really marked effect in a species like the rat because of this proliferation of peroxisomes. So what we're suggesting is that when the mother has been fed a low protein diet, we then get the offspring, and as they get into later life, compared to the controls, they had a higher level of SREB1C. That increased the expression of the lipogenic genes, and we showed an increase in the mRNA for both ACC and FAS. That leads to an increase in the amount of fatty acid being made, increase in the amount of hepatic triglyceride, an increase in VLDL secretion, so the plasma triglyceride goes up as well. On the other hand, PPAR alpha is down in these offspring, therefore there's a reduction in fatty acid oxidation, and again we showed a reduction in some of the target genes, so it wasn't just the transcription factor. You're oxidizing less, therefore this can accumulate and that can accumulate. So from both arms, an increase in synthesis, a reduction in oxidation, we believe that's where the extra triglyceride might be coming from, which is accumulating in the liver and in the VLDL. So the summary of that study is that in normal rats, aging is associated with changes in hepatic lipid metabolism which prevent the accumulation of lipid. It appears that perhaps there's a need as we get older to make less fat and perhaps oxidize more to prevent it accumulating. Certainly in terms of the gene expression at least, that's what the data suggested. But in the offspring of low protein fed mothers, this program seems to be disrupted completely. It results in non-alcoholic fatty liver disease, hypertriglyceridemia, and appears to be associated with altered expression of the transcription factors SREP1C and PPAR alpha. But how? And this is the big question, the question that everybody is trying to get to first at the moment. A lot of work and some positive results suggesting that there are changes in maternal endocrine status that have an impact on the development of the offspring. Glucocorticoids have been studied and a whole range of factors associated with kidney function. Retarded organ development. We know that very often there is a reduced number of beta cells in the pancreas of offspring from mothers which have been fed either a nutrient deficient or a protein deficient diet. That could lead to insulin resistance. We know that there's a reduction in the number of nephrons, and that, my colleagues in Nottingham have been involved in a lot of that work, potentially leading to an increased risk of hypertension. The alternative is a potential role of epigenetics. I'm sure you all know what epigenetics is, and I apologize for resorting to Wikipedia, but sometimes it's the best possible place to go, despite what I tell my students. So according to Wikipedia, epigenetics is a term used which refers to changes in gene expression that are stable over rounds of cell division, sometimes between generations, but do not involve changes in underlying DNA sequence. And that's important. We're not changing the sequence of bases. We're changing chemical modifications to the actual DNA itself. The molecular basis of epigenetics involves modifications to the DNA and the chromatin proteins which are associated with it. So it is a change in our DNA programming, if you like, but we're not changing the bases. It's not a genetic change in that sense. We're changing other molecules which associate with the DNA, which change our activity in transcribing that DNA. And probably the most studied change, and the one which has been concentrated on most, is DNA methylation. Basically, in order to be able to read the DNA, to produce the messenger RNA, the transcription, you need a fairly loose structure. You need it to be the enzymes which are involved in the transcription to be able to get at the DNA base sequence. If you methylate, if you put a methyl group in certain places along that DNA, it changes the whole physical structure, and it packs it together very carefully like we see here. And because it gets packed together like that, the enzymes involved in transcription can't get in to actually start transcribing the DNA. So methylation is used frequently in biology to turn a gene off. And obviously we do that all the time. If you want to produce a hand, you need to have some genes being turned on to produce that hand, and you need lots of genes turned off to produce it. And methylation is one of the ways in which we do this. If that methylation happens to be in the promoter region of a gene, then you can see that it can actually affect the regulation of that gene. Here we have our gene, here we have the promoter, here we have some sites where transcription factors might bind. They bind to that, they stimulate the gene, you get lots more RNA being produced, you get lots more protein being produced from that RNA. If we methylate this site, the transcription factor that wants to bind can no longer bind to there anymore. Therefore, you turn off the gene and you produce less of it. So immediately you see a potential mechanism whereby our fetal programming might be working. If we happen to have methylated areas on the promoter of certain genes, does that stop them being activated and then thereby change the whole of the program? I can't answer that for definite in terms of our experiment, but there is some interesting evidence which has just emerged. And it relates to yet another transcription factor. And this one's called the liver X receptor. And this was not our experiment, this is from the literature, an experiment where mice were exposed to a protein restriction during pregnancy, the same as ours, 9 versus 18%. And then they did what we call a methylation microarray to find out where all of the differences were in the methylation of the genome in the liver of these animals. They found 204 gene promoters where there was differential methylation, and one of them happened to be the liver X receptor. It was hypermethylated. More importantly, it was hypermethylated. Some of that methylation was actually within the promoter. Now, LXR is another nuclear receptor, as I say, and is actively involved in the control of cholesterol and fatty acid metabolism. And as you'll see in a moment, more importantly, it's specifically involved in the regulation of SREB1C. So the mRNA level of LXR was reduced by 32% within the fetal livers. So that methylation reduced the production of LXR itself as well, of LXR. And the target genes, some of the target genes were reduced as well. Many years ago now, we actually sequenced the human SREB1C promoter, and we identified potential nuclear transcription factor binding sites. And right in the middle of it, as you see, we have 2 sites which are specifically sites where LXR would normally bind. Various tissue culture experiments and what have you, not just by us, but by many other people, have shown that one of the major regulators of SREB1C is LXR. So, if low protein feeding reduces the expression of LXR by methylating the promoter, there's less LXR around, therefore there's less LXR to bind to SREB1C, therefore you reduce the amount of SREB1C. Then, further on, you will then reduce lipogenesis, you'll reduce fatty acid synthase, acetyl-CoA carboxylase and everything, and so you've got a cascade of different effects. And if you remember, that's exactly what we saw early in life in our offspring. They actually had reduced SREB1C. What we don't understand is why 18 months later, all of a sudden that programming goes away and they've actually got increased SREB1C. But we're beginning just to get maybe an insight into some of the molecular mechanisms by which we're seeing our effects. So methylation of LXR promoter is increased, decreased expression of LXR, decreased expression of SREB1C, decreased expression of all of our lipogenic enzymes, making less fatty acid. But why is it reversed later in life? No. Sorry, I can't go any further at the moment. I just haven't got an answer. So coming to an end, what nutrients are important? Vast number of studies, early studies, were on either, as I say, global nutrient deficiency or protein restriction. However, there is some evidence for micronutrients. Colleagues in Scotland, the Rowett, have looked at iron in particular, and iron deficiency seems to program a number of metabolic diseases. And there's an increasing evidence that B vitamins are involved. And I'll bring that back to what we were hearing very earlier about the difference between Asians and Caucasians. In that, I went to a talk a few weeks ago by a fairly eminent professor from India who was looking at this problem. And his theory is that mothers in India are being supplemented specifically with folate. There's a big program of folate supplementation going on, but this is being done against a background of a very poor diet. And in particular, they have very low levels of vitamin B12 in their diet. His theory, whether it turns out to be true or not, is that this mismatch between the amount of folate and the amount of B12 in their diet is having a major impact on fetal programming, and they're programming this increased susceptibility to obesity and insulin resistance. And I haven't got time to go through all his evidence, but it was fairly persuasive that this might be one of the effects. Most recently, and perhaps most relevantly for most of us, is the question of overnutrition. Unfortunately, again in the Western, the developed world, we're seeing more and more mothers who are actually obese rather than suffering from undernutrition. What impact is that having on the potential health of the offspring? Again, the evidence is emerging that, again, overnutrition may be equally as bad as undernutrition, and you may be getting susceptibility, particularly again, to insulin resistance and diabetes in the offspring. So, I said I was going to look at what, why, and how. What? There is now considerable evidence, both human and animal, that maternal nutrition can have a significant impact on the development of chronic disease later in life, in the offspring's life. Why? It's probably a result of mechanisms to try and ensure that that fetus survives the pregnancy and is born relatively healthy, but not predicting what's going to happen to it in 30, 40, 50, 60 years' time. How? We're nowhere near knowing the exact answer to this, but it could be that epigenetic changes, it could be that specific changes in methylation are actually having an effect. With my particular gene of interest, which was SREB1C, it looks like there might be emerging a pattern. How that is specific, why there's only certain genes targeted, is a massive question. To finish off just by saying as well, my colleague Simon Langley-Evans has been going back and back and back trying to find what the earliest event is, trying to get early and earlier embryos to see whether they can see changes. And quite excitingly, his latest changes are changes in proteins which are involved in regulating the cell cycle. So it may be those very early divisions which are going on in the embryo are the first thing which are affected. You can imagine if you just change one division, if you delay one division in the offspring, then you've changed the whole programming of its development, because every time it doubles, it gets bigger. So it may be that there are changes very early on which lead to all of these other changes that we're talking about. I don't know. I think some of the fetal programming world get a bit carried away. They think it's the be-all and end-all. They think it's everything. It is one factor. And I think it's very important to remember that in terms of whether you're going to have a heart attack, whether you're going to get cancer, whether you're going to get diabetes, your own genetic predisposition is going to be important. Yes, your prenatal influences, what your mother ate, potentially even what your father ate, and I haven't gone into that side of programming, but there's some evidence that that might be important, may have an effect. But equally, what we were talking about earlier, postnatal influence, what you eat during your lifetime is also important. Remember, the mice that ate the low-fat diet didn't get heart disease at all, so it's only if you challenge yourself that you're going to have an effect. Leave it there. Those are some of my colleagues in Nottingham who are involved in the whole process. Thank you very much. I'm more than happy to answer some questions.
Are there any questions? Just one from me. Okay. In the previous slide, would you care to propose percentages of importance?
Like these, all right. 40 questions. Don't quote me, whatever you do. I still think that there is a strong genetic component. We all know that. I mean, we're not talking about individual polymorphisms. We're talking about people who are relatively protected from heart disease, those who are. We all know somebody whose granddad had a fried breakfast every day, who smoked 20 cigarettes a day and lived till he was 90 because he was genetically very robust. So that has to be a major influence. If you don't challenge the system, you're not going to develop heart disease. But I don't know, 40, 40, 20, 45, 45, 10, I would put it in the influence of those. There are people who disagree with me, though, including David Parker himself.
Okay, thank you.
Okay? Yeah. So you got a question?
Yeah, just maybe one question about the thrifty genes hypothesis. I think I agree that it's not preparing the baby for an environment, you know, where it's got lack of food and he's got to have thrifty genes. I do think that the thrifty genes were an evolutionary pressure in our pre-ancestors, and when there was not enough food available. But that's a different perspective.
I agree entirely with the thrifty gene hypothesis. I believe that that's why some populations have survived. You look like the Pima Indians in America who were doing really well. But I was talking about thrifty phenotype rather than thrifty gene, and there's an important distinction which lots of people... I don't believe that baby's sitting in there predicting, oh, I'm going to have a hard life, I better change all my gene expression. It just doesn't work for me. But in terms of evolution and developing thrifty genes, I agree with you completely.
Any more questions? Nope.